Good morning, everyone. Thank you all for joining the third and last session of this training series on essential GIBA concepts and techniques. So I'm Alex, one of the colleagues of the GIBA network. I'm joined today by Joanna Neves and Fran Denk, who will be giving you the lectures on the immune portion of this course. So just as a bit of an introduction, just to keep you aware, so you probably realize you have the mics and cameras off, so this is really just to avoid having any technical issues or interference while the speakers are talking. However, we do encourage you to use a chat function to ask questions. So the way this will work, each speaker will give a 40-minute talk, and then at the end, I'll read the questions out that were written in the chat, and we'll have a discussion there. If you happen to see a question written by someone else that you are interested in hearing the answer to, please do add a thumbs up. I'll try to prioritize questions that have more thumbs up in the chat to ensure that we have enough time to address those. So in terms of the recording, so the session will be recorded. I've had a few people asking if they'll be made available, how they'll be made available. We'll need just a bit of time to edit them slightly and break them down to individual talks. But then once that is done, we'll make them available on the YouTube channel. So don't worry, you'll be able to look back at the talks and consult them there. So we're going to get started first with Fran Denk. So Fran Denk is an associate professor at King's College London, also one of the co-leads of the UK GIBA Network Plus, and her work investigates the peripheral nervous system specifically and its role in chronic pain, with the particular focus on why pain persists over time. So her work examines neuro-immune interactions and epigenetic mechanisms, and she uses various different models, such as transgenic models and high-throughput molecular techniques to better understand pain biology. So Fran is going to give us an overview of the peripheral nervous system from an immune perspective. So over to you, Fran, whenever you're ready.
Thank you so much, Alex. Welcome, everybody. Thank you so much for joining this morning. And I hope you'll bear with me, because even though this is badged as the immune session, I think it's really useful and important that we keep considering that for the immune system in the gut to actually sort of communicate, reach our brain, there's various ways. One way is, of course, systemically, and now there's also some newer data that there might be homing T cells somewhere in the dura of the brain and so on. But conventionally, most of the communication from your immune system in the gut to your brain will happen through afferent peripheral nerves. So peripheral nerves that send information up into the brain. And so I thought it could be really useful to go through the basic anatomy of these afferent peripheral nervous systems, because it turns out they're quite complicated. So that is how we're going to get started. We're going to start with some relatively basic anatomy, and then I'll give you some examples or some ideas of how we study these different nerves and what is known, and kind of resources where you can look up what we know about these nerves. And then finally, I give you 3 examples, very recent literature of sort of the cutting edge of what we're sort of discovering in this space about how the immune system communicates with these nerves, about the immune status, about host defense, about inflammation. So firstly, we have extrinsic, and I'm going to use these terms, extrinsic meaning outside, so nerves that leave the gut, as opposed to the enteric nervous system, which would be intrinsic. So we have a lot of extrinsic sensory innervation. So these are sensory nerves that leave the gut, and they end up through various nerves. So like through the pelvic nerve, they leave the gut. They leave the gut through celiac and splanchnic nerves. And then also they leave the gut through the vagus nerve. And I'm going to go into a bit more detail about this extrinsic sensory information. And these are sensory nerves that carry information up from the gut into the brain, so afferent as opposed to efferent. When we talk about extrinsic sensory information, these nerves that run through the pelvic and splanchnic nerves, they actually have their cell bodies really far away from the gut in so-called dorsal root ganglia, sort of balls, collection of cell bodies that sit basically underneath your vertebra, very close to your vertebra of your spinal cord. You have about 40 of these in your body. And so they are pseudo-unipolar cells. That means their cell bodies are in these ganglia and the dorsal root ganglia. And then they send an axon into the periphery, and this here it's the images from a finger, but it would be into the gut, and then they send the other end into the spinal cord. I want to note at this point that even though when they leave the gut, going back here for a picture, so they would leave the gut through the pelvic nerve, but they don't actually, and they also pass through things like pelvic ganglia, so like close by to pelvic ganglia. The pelvic ganglia are the efferent information, autonomic nerves, but they pass through all of this to reach their cell bodies. It's like one long nerve basically that runs within the pelvic nerve to directly hook up information from your pelvis to the spinal cord, and same for the celiac and splanchnic pathways. So here again, we have a bit more detail on this. So they're long pseudounipolar cells. You have here, for example, is of different types. So you have peptidergic types. They make this peptidergic neurotransmitter called CGRP. And here is the cell body, and it sends information into the dorsal horn of the spinal cord, but it then also goes all the way into your gut walls. So here is the gut represented. You have your gut wall, and then you have your lamina propria. This is where a lot of the immune cells are, and then you have the different enteric nervous system plexuses. So this is where these round things are supposed to represent the ecteric neuron ganglia. So they sit in little balls of cells, and enteric nerves are entirely in the gut, right? If you take the gut tissue, you can take out the enteric nerves. You can look at them, but if you take the gut tissue, you cannot take out the sensory nerves. Their cell bodies are somewhere completely different. And if you do, for example, RNA sequencing, the signal coming from these tiny axons is too little for us to currently pick up with. We don't have the sensitivity to really pick up on this signal with say, conventional single cell sequencing technologies, for example. So you can see that in this case, this is a review about CGRP positive and negative sensory afferents, and they were studied in terms of where they terminate. But you can see that both of them terminate both all the way directly to the lamina propria. So they're really, really, really close to your empty space in the gut. So you can really sense what is going on there. But they also terminate in the higher layers. Here the myenteric plexus, for example, and speak to enteric neurons as well. And once more, I'm just driving home this point because even for myself, it can be a little bit complicated sometimes to remember. They then leave the different bits of your gut in different nerves. So for example, the large intestine then will be served by the pelvic nerve. And then these are a representation of your different dorsal root ganglia. So if you're interested in, rectum or large intestinal sort of sensations, you have to look at the pelvic nerve and you have to look at these ganglia, sacral ganglia. Whereas if you're interested in the small intestine, you have to study the splanchnic nerve, because this is where the information ultimately ends up in the cell bodies here in L1 to L6 lumbar dorsal root ganglia. And then the stomach, once again, is somewhere else. It leaves somewhere else. So the information is in different places. And so you have to really be familiar with the anatomy to make sure that you're studying the right nerves for the right question. So that's the extrinsic sensory afferents with their cell bodies in the dorsal root ganglia. There is another big nerve that carries afferent information from the gut, including and also other visceral organs, and that is the vagus nerve. So the vagus nerve, of course, you will be familiar, also carries information from the brain to the gut, but it also carries information from the gut to the brain. So it's mixed. It carries both afferent and efferent information. And the afferent information from the vagus nerve, again, has the cell body very far away from the gut in this sort of nodose ganglia. What is nice about the vagus afferent information, it's all in one place. There's just a pair of ganglia, nodose ganglia, left and right to your brainstem. Once again, pseudounipolar, you have cells that are really long. They send one end into the intestine and then another end into the nucleus of the solitary tract. It's called NTS here in your brainstem. So directly communicating, or rather directly receiving signals from your intestine to the brainstem. But so if you're interested in those afferents, you only have to dissect out two ganglia. If you're interested in sensory nerves, the other sensory nerves that I'm interested in, you have to dissect out a lot more ganglia that are distributed along the spinal cord. Yeah, and the vagal efferents would come from the dorsal motor nucleus of the vagus, so they come from a different place. Vagal afferents receive information once again from the lamina propria directly. So these extrinsic sensory informations, they really receive information right at the cusp of the gut. You have the lumen, and they're sitting right behind in the lamina propria, also where all the immune cells sit. So they're really, really attuned to what is going on, and they can report everything immediately to your spinal cord and brain. And once again, afferent vagal nerves also synapse with enteric neurons, so they can also indirectly hear what is going on. Right. So complex anatomical picture. I hope I have not put you off with that, but I think it's important to bear that in mind. Now, where do you actually find out what is in these kinds of neurons? Like, where can you find out, is your favorite gene there? Are you, should you be looking at these? If you're interested in neuro-immune communications in the gut, how are these communications happening? It's usually useful to know which cell expresses what gene. And luckily, these days, there's lots of useful gene expression data resources, like fast lookups just on the internet. Like this website here is from the Karolinska Institute. These are the people that some of these teams basically co-invented single-cell RNA sequencing around Sten Linnerson and many others. And so they have a very nice, this is now mouse, but still very useful classical website that contains single-cell RNA sequencing data of lots and lots and lots of different neuron subtypes in the brain, the spinal cord, but also in enteric, sympathetic, and sensory afferent ganglia. So here you can look up, here, for example, I'm looking up the CGRP gene, this very well-known peptidic neurotransmitter, because CGRP is one of the main ways in which sensory nerves can actually influence the immune cells around them, and can also influence the blood supply. So they can cause, when you have swelling and redness during inflammation, that's because your sensory nerves make CGRP and the blood vessels sort of expand. And you can see here where there's blue, it shows the CGRP, likelihood of CGRP expression. With this website, the bluer something is, the more likely it is to be expressed. So you can see that there is a subset of sensory nerves that likely expresses CGRP. You will also see that sympathetic nerves, for example, do not make CGRP, nor do enteric neurons, and pretty much no other nerve, in the nervous system makes CGRP. So the CGRP comes almost exclusively from these sensory afferents. And here you can add genes. So here I'm adding tubulins, a kind of neuronal marker, yeah. And I'm adding, for instance, TH. You may be familiar with TH. Tyrosine hydroxylase is often used as a marker for sympathetic nerves. Bear in mind that some sensory neuron populations also express tyrosine hydroxylase, although primarily in the skin, I believe. Well, they see low threshold mechanoreceptors. So perhaps also in the gut. I'm not too sure. But in any case, you can look up different genes and where all they are expressed on the press of a button. The same thing exists now for human dorsal root ganglia. There is a couple of websites I would recommend there. They're really nice. So Genentech, for example, has a cross-species atlas where they compare human dorsal root ganglia with mouse and guinea pig and monkey. And then we also have now from the US, there's some consortia that are really, really amazing at sequencing many, many hundreds of human DRG. So I think they're up to 127 human DRG. This picture is CGRP expression across these 127 human DRG and the different kind of subpopulations that exist there. What is notable is that none of these resources, kind of overall resources, these are just sensory neurons in general. They're not the sensory neurons that innervate the gut, right? Because how would you know? They're all mixed together in the ganglia. Some of the sensory neurons in these ganglia come from the gut, some will come from the leg, some will come from, the skin. So at the moment, especially when you're taking ganglia from human, you cannot really know which is which. You just know overall in this, say, lumbar 3 ganglion, some of the neurons will project to the gut and some of the neurons will project elsewhere, and this is what they express in totality. There's also websites here, for example, where you can start looking to see what happens if you damage nerves. My lab is very interested in nerve damage. So here, for example, we've got a website where you can look up what happens to sensory nerves when you cut into them. I mean, this is quite an extreme damage, but I wanted to show you this because what you can see is that one of the genes that is the most upregulated genes in this context is CSF1. The immunologist among you will recognize CSF1 is the main macrophage monocyte chemoattractant gene. So sensory nerves, when they're damaged or distressed, will very quickly call on the immune system for help and will say, come help me do something. And so you'll get a big immune reaction and you'll get resident macrophages in the various tissues come and move towards the nerve and try and somehow help, which sometimes can have unwanted side effects as well. There is one paper that I'm aware of so far where they've actually labeled, and obviously this is something you can only do in the mouse, where they've actually injected a retrograde tracer into the colon of a mouse. And so then you can, that will be carried up via the lumbar splanchnic in the pelvic nerves into the cell bodies of these neurons in the respective ganglia. Apologies. And then you can do single cell sequencing on this. So this is the one paper that I'm familiar with from you and Sir John Smith's lab in Cambridge, where you can actually look specifically at what the nerves express that project to the gut only. The good news in a way for looking up information even right now is that currently it seems to be, according to our current knowledge, the main thing that we find is that peptidogenic afferents predominate in the gut, but otherwise there are no gut-specific populations. So any kind of sensory ganglia or any organ in your body, when you look at DRG ganglion neurons, most types of DRG neurons project to most organs with some perhaps minor differences. With the vagus, this has also been done in the mouse. And so again, with the vagus, you can do this quite nicely because you have, as I said, you only have to dissect out 2 nodose ganglia. And the difference between the vagus afferent information and the DRG afferent information, that actually in the vagus nerve, it seems that it actually is organized quite nicely in terms of organ of origin. So it's very clear that the nerves that project to the pancreas, or rather project from the pancreas and carry afferent information into the nodose ganglia, they're different and they're in different places. And this was a very sort of seminal paper that showed this very nicely with some like really cool constructs in mouse. And once again, these are data that I think you can download. This is maybe slightly harder to access, but you certainly can find, look up your favorite gene with a bit of extra bioinformatics in R. All right, so I've talked about the anatomy. I've talked about where you could look up what these genes express and what these different nerve populations express, at least at mRNA level. And now let's talk about how do we study these different nerve populations? What are the options? If you're interested in neuroimmune interactions, how can you actually look at the neurons? So primary culture is one of the main ways in which they were studied. The nice thing about peripheral afferent neurons compared to cortical neurons is that if you take them out of an animal and you put them in a dish, they actually survive. They grow quite nicely. Here you have an example of rodent DRG neurons. These big round things, those are your sensory nerves. And then some other non-neuronal cells are also in these dishes. And here you can look, if you enable them with immunostaining, this is tubulin. You can see they actually grow nice little neurites in this case, and they start branching out and acting like real neurons. You can do the same from human postmortem donors. Here is an example of a human postmortem donor culture. And once again, you can show that they respond to the things they're meant to respond to. Your sensory nerves are meant to respond to things like histamine, for example, the molecule that causes itch. And here, these scientists put histamine on these nerves, and you can see a lot of action potential firing. So that's certainly something that one can do. Of course, having a system set up where you get a human post-mortem donations is not easy, and they don't survive forever. You can keep them in a dish, most of these, for maybe 5 days ideally max, but sometimes a bit longer, maybe 2 to 4 weeks, but it's sort of time limited. The other thing that's important to note is that these primary culture cells, they contain a lot of non-neuronal cells because you're taking them out of the ganglia. Remember the cell body in the ganglia, and the ganglia contain fibroblasts and glial cells and Schwann cells and all sorts of other cells. And they'll all be in your dish. And right now, I know of only one way to definitively remove all of the supporting cells. This is a magnetic sorting protocol that works for mouse nerves because the antibodies are mouse specific. So there is no way at the moment to do this in rat and human. So basically, if you use these primary cultures, can be very useful, but bear in mind that you're not just studying the communication between, let's say, your favorite macrophage and a sensory nerve. You're often studying then the communication between your favorite macrophage, a sensory nerve, mediated by fibroblasts, glial cells, and all sorts of other cell types. Nodose ganglion cells also grow very nicely in a dish. Same caveats apply. You will have non-neuronal cells here, but so you can study it beautifully. And if you're interested in the electrical activity, once again, this is a patch clamp experiment. This was actually a very famous study where they showed that endocrine cells can interact with vagus nerves directly. So they're patching these vagus nerves, and then they apply things, and they can show that they respond as expected. If you want to study human systems, besides the post-mortem option, and if you want sort of more cleaner nerves, one option is to grow these sensory nerves from stem cells. Our lab does this quite a lot as well. So these are some stem cell-derived sensory neurons. Of course, they're good for certain questions, but not for others, because they're not the perfect sensory neurons. They're slightly different from the ones that exist in vivo, but they're nowadays, it's quite well described, they're sequenced, we know exactly what is in there and what is not in there. And currently most people use a version of a protocol called Chambers et al. So we have a good handle on what we're making in these dishes. And they tend to be, if the differentiation goes well, relatively clean. So this is another way of potentially studying sensory neurons without non-neuronal contamination, if you will, although it has other disadvantages. Here, for example, what we showed is that you can activate these nerves with a cytokine called LIF. So these are human sensory neurons, and you plate them on an array with electrodes where you record their activity, and they fire more and more action potentials as you heat up the plate. That makes sense because sensory nerves are meant to tell you when it's getting hot. This increase in firing is exacerbated when you add the cytokine LIF. So they're becoming more excitable because it's something that they don't like. So again, these are fairly well described systems and can be used if you're interested, an immunologist interested in finding out what the neurons are happening. All of these techniques exist. Another big thing that often gets used is to identify them functionally in vivo and studying them. So here, for example, you use, most often nowadays, people use a calcium imaging. So they label them with a calcium sensor. So whenever calcium changes, the color of the cell changes, and you can record that with a confocal microscope or a 2-photon microscope. And so here, for example, are nerves that are responding to a gut colon extension. And similarly, in the vagus nerve, this is also possible. You can, these are mice that are alive but terminally anesthetized, and you can once again record their calcium signals directly as a proxy for their electrical activity. If you want direct electrical activity, there are a couple of labs in the world that do this. It is, again, very specialized knowledge. You can record from the extrinsic nerve bundles that run alongside mesenteric blood vessels. So this is actually an image from human appendix where this is feasible. And so here is a paper where there is a group in Australia that does this a lot. And so again, they record from these nerves. Here the LSN stands for lumbar-splanchnic nerve. So they record from these nerves, and while they sort of extend the gut, these are ex vivo assays. So it's like in some kind of bath. And then you can essentially figure out which sensory nerves are mechanically sensitive, insensitive, and so on and so forth. Here, for example, is a nociceptor, meaning it's a sensory neuron that picks up on noxious things in your gut wall. And similarly, here is another nociceptor that you can show that they become sensitized. They become more responsive to bad things. So here you would, they're recording from a particular neuron that normally doesn't respond to mechanical stimulation. And then they add something toxic, something bad called bradykinin. It responds to the bradykinin because it doesn't like it. And then after that, it responds to mechanical stimulation. It has become sensitized. It's now, freaked out and will say something's not wrong. There's inflammation in the environment. All right. So in the last 10 minutes, I wanted to talk you through now to some of the studies, like how have these techniques been used now to uncover how the immune system talks to the sensory nerves, how the immune system relays that there's inflammation, that there's a problem in the gut. And I've chosen 3 studies from 3 very sort of cutting edge teams. They are all mouse. Again, doing this in human is much more complicated, but I think hopefully they'll be give you a sort of a flavor of what we know and what we don't yet know. So the first example is an example that shows how inflammation causes afferent sensory neuron activation. It's an example from David Ginty's lab in the US and colleagues. David Ginty's lab always does very beautiful tools. They make tons of mouse lines where they label lots of different sensory neuron populations and do very systematic studies. So here, for example, they characterized the gut innervation patterns of DRG neurons, and they made these fancy mouse lines where they had different Cree drivers, and so they can look at colored, sort of, they look at the afferents are colored in different ways, and they can look exactly where they end up. So, for example, this particular sensory neuron marker, BMP1B, sends afferents into the myenteric plexus, less so in the submucosal plexus. And then they basically do this for different sensory neuron markers, so TrkB, TH, and so on. And so they can define like where different types of sensory neurons go and where exactly which layers in the gut they end up. So we're still a little bit at the basic anatomy level, right? Because we know grossly they end up in these different layers, but are there sort of differences with the different molecular subtypes? That's sort of what people are working out at the moment. And so this is what they have come up with. There's beautiful results, like, they've kind of shown, okay, so you remember you have I told you the different plexuses, enteric neuron plexuses in the myenteric plexus and the submucosal plexus. And so the extrinsic sensory neurons end up in slightly different places. So for example, ADRA2A, positive extrinsic sensory neurons add up in the myenteric plexus and the submucosal plexus, whereas BMPR1B, positive sensory neurons end up primarily in the myenteric plexus, and so on and so forth. And they have slightly different ending shapes, and that might also affect their function, and so forth. So that is being studied at the moment. And in fact, what they showed in this paper is that BMPR1B neurons show relatively selective sensitivity for high pressure colonic distension. So here again, they have this in vivo imaging technique that I described briefly. They can extend the colon with different pressures, and then they record to what extent these different sensory neuron populations respond. And you can see that TH positive neurons, for example, respond already at 60 bars. They respond quite quickly, so they don't really distinguish between the things that are like bad noxious pressure and sort of regular pressure, whereas these BMPR1B neurons almost exclusively fire at the highest level of pressure. So they're like really there to say, okay, now it's going really badly, you're really damaging, big damage, big problem. And that's what these nerves are specializing in. And they also show that these neurons are actually necessary for pain upon colonic inflammation. So here they use this, it's called DSS model. It's a model of colonic inflammation. It's quite crude. And you can see you inject basically some toxic substance into the gut, and you can see that everything becomes completely destroyed and inflamed. Here is the histology of a normal gut, and then here it becomes destroyed, and then you can see how an animal reacts to this. And if you delete these BMPR1B nerves, it turns out that they don't feel the pain of this happening, because these nerves are the ones that say you should be in pain right now. The second paper I want to mention briefly shows, or the topic is about afferent sensory neurons impacting immune cell function. So interestingly, even though we're talking about afferent sensory nerves, so sensory nerves that normally carry information from the organ to the body, it turns out that locally, actually, they also impact the immune system. They can talk back to the immune system and determine what happens. And in the pain field, the person who's been studying a lot of this and has been really pioneering in this is Isaac Chu from Harvard University. And his team, they've looked a lot at how sensory neurons can sort of talk back to the immune system. And this was one of the papers where they looked at this in the gut, and they turned off a specific sensory neuron populations with a technique called chemogenetics. I don't know whether you're familiar with this. It's sort of a technique neuroscientists use a lot. It's a designer receptor, so basically a receptor that doesn't really exist normally in us or in any organism. But so it has a ligand that is artificial. CNO is the ligand. It doesn't really exist. And so if you inject this now into this mouse, it will specifically activate this designer receptor that you've inserted into the mouse. And then you can make the system Cre-dependent so that the designer receptor is only expressed in the cell types that you're interested in. And so they did this again in all these different mice lines. I mean, it's an enormous amount of work. And then they show where all of these expressions are. So they use different Cre driver lines to mark different subpopulations again. So here, in fact, they also did this in nodose ganglion. They looked at DRG. They looked at enteric neurons in the myenteric plexus. And they had studied lots of these different populations. And then they basically said, okay, so now if we shut off all these different populations, what's the effect on immune cells in the gut? So just like a systematic investigation. And then they also went along the length of the gut. So immune cells in the colon, immune cells in the cecum, immune cells in the ileum. And this table looks quite complicated, but basically most of the time when you shut off all these sensory neurons, nothing happens. But there are a few populations that are affected, and basically the color indicates that there has been a change. Blue means there has been a significant downregulation. Red means there has been a significant upregulation. Bear in mind that obviously they're doing an enormous amount of comparisons here, and I don't think they have multiple comparisons corrected, so there's still sort of a certain amount of uncertainty with this experiment alone, but it's quite interesting to see. It's been done sort of systematically, and there are some changes, and in fact the biggest change they saw and that they then followed up on with many more experiments was that you can affect T cells, especially regulatory T cells, by manipulating afferent sensory neurons. And they did several experiments to show that this is not to do with nodose ganglia. And this is one of the experiments. So again, they destroy either DRG sensory neurons or nodose ganglion neurons. And then they see what's the effect on FOXP3 T cells. And here you see this is normally your level of FOXP3 T cells in the gut. And then if you destroy your sensory nerves, they're just fewer FOXP3 T cells. Whereas if you destroy your nodose ganglion nerves, it has no effect. So it seems to be that having sensory nerves there in the gut wall, in the lamina propria, is relevant, will to some extent affect your regulatory T cell number in the gut. And that sort of, again, from an evolutionary perspective, is not sort of relatively logical that sensory nerves and the immune system are sort of closely coupled functionally, because of course both of them are primarily responsible for host defense. And here, by the way, there's beautiful images where they show these sensory nerves really closely associating with these FOXP3 T cells in red. And in fact, these are CGRP positive afferents. I told you how CGRP is this neurotransmitter that talks to the immune cells the most. And once again, here, indeed, they show that CGRP is really relevant in this context as well. If you knock out the CGRP receptor, you don't get the same kinds of effects. All right, we're almost done. The final paper I wanted to mention briefly is about afferent vagal neuron activation. So again, here we're actually quite at sort of doing quite, if you will, basic experiments. They're very complicated, very, very technically sophisticated experiments, but the questions we're answering are still quite basic. So for example, Eric Chang and colleagues showed recently that inflammation does indeed cause afferent vagal neuron activation. And they showed this directly with this in vivo imaging. So they put cytokines on on the vagus nerve, and then they can show that the vagal nerves can respond to these cytokines. And indeed, they showed that colitis, once again with this DSS model, can cause vagal neuron activation in the nodus ganglia. So they measure directly that the vagus nerve will fire more if there is inflammation in your gut. That's what the graph shows on the left. And so we're sort of slowly building up the body of evidence to show that there is an important connection and important information flow from the gut via the vagus nerve. So in conclusion, then, what I really want you to take away from this is that extrinsic afferent peripheral gut innervation is very complicated, right? Always make sure that you know the anatomical realities before you delve into certain questions. It's unlikely some random microbiota in the lumen has to somehow, how does it reach the brain? How does it affect the brain? And there will be many different anatomical connections, and it's important to be aware of the sort of possibilities. I also want to make clear that there's still lots of known unknowns and many questions for research, especially in humans. So that's good news for us scientists. Many questions still outstanding, many exciting topics to still explore. And then, of course, remember that I'm only showing you like half of the peripheral neuron innervation here. I was talking about afferent information, so information coming from the gut to the brain. But of course, there is an enormous amount of extrinsic efferent information via sympathetic and parasympathetic and vagal nerves. So the brain talking back and saying, do this, do that, to the immune system. That's another really big piece of the picture. And that's all I was going to share with you today. Thank you so much. And I'm ready for questions.
Great. Thanks, Fran. That was a very nice overview. Lots to dig into. So yeah, do please add your questions to Fran. We have already one here. So thank you for this resourceful presentation. I have a question about whether approaches like single cell sequencing and iPS-derived neurons used to study pain could help us understand how gut nerves become hypersensitive in conditions like IBS or IBD. They further expand in chronic pain, peripheral nerves become sensitized. Could a leaky gut allow immune molecules to reach gut nerves and trigger that kind of sensitization?
Yeah, absolutely. Very good question. And in fact, that is being studied. So when I showed you, I went over it slightly fast, my apologies, that study where they looked at what bradykinin does to the nerve, the sensory nerve in the gut. That is pretty much what people believe is happening. And there is quite a lot of evidence, actually, at least in mouse, that is what's happening. So when you have IBS or IBD, when you have colitis, what's happening is that the inflammation will cause spontaneous activity in these sensory nerves. So normally these sensory nerves are silent. They don't have any tonic background activity. So they start firing, and that will register as pain. But the other thing that happens is that indeed they're being activated by substances like pro-inflammatory cytokines, things like bradykinin, serotonin as well can do these things, prostaglandin, the thing that we block with ibuprofen. Those substances will sensitize the nerve. And what is meant by that is that a nerve that previously doesn't respond to touch, for example, once sensitized will respond to touch. So these are indeed the reasons why IBD and IBS are so painful and uncomfortable. It's because these sensory nerves are saying something's not right in our environment. There's substances, there's factors here, there's stimulation here that is noxious, that is not good for us. I hope that answered your question.
Yeah.
Sorry, just to say, I have to add, maybe in stem cells, the way we study this is that we know once we know the factors, you can put them on your stem cell-derived sensory neurons and you can see whether they fire more, like with LIF, for example. The reason we added LIF is because we found in another painful condition that this is a substance that might make them angry, so to speak. And so that's why we put it onto the nerves to kind of really show that indeed this is now the substance that causes the hyperexcitability and therefore potentially the pain.
So I guess a follow-up question from me. So in terms of thinking about therapeutic implications of that, so are people trying to look at how targeting those sensory cells and neurons to reduce pain sensitization in people, for instance, have IBD, IBS? Is that one of the goals in the field?
Exactly. And so there's basically three ways that people think about this. The first way is that you indeed just try and take away the substances. You try and take away the inflammation. So, reduce the inflammation in IBD and then the pain would also go. That's the idea.
Yeah.
The second thing is that if you can't do that, you try and block the receptors for these substances on the sensory neurons, or try and stop the sensory neurons from firing. So actually, the acute pain medication that just got approved in the United States for acute postoperative pain, that blocks an ion channel that just stops action potentials in these nerves. So that can be analgesic. And then the third factor, especially in IBS, there's a lot of thought about that maybe not all of it comes from the periphery, but that also once the nerve, and I didn't talk about this because this is pain specific, once the nerve sends the signal to the spinal cord sends it up to the brain, and there's lots of modulation happening at that level, and the brain is what generates the pain percept, and so the brain has a lot of control, and the brain these circuits can sometimes go wrong as well. And so then other drug interventions happen at the spinal cord. So for instance, antidepressants are oddly often prescribed against pain, and people get confused. The reason they get prescribed against pain is because they tap into these spinal cord circuits and try and sort of regulate them again properly so that the information doesn't get scrambled when it comes in, basically. So there's these options as well.
That's interesting. Yeah, so it's interesting because there's a lot of talk about also the role of medication in the gut microbiome composition, and one of the strongest types of medication that have been involved in changing the microbiome is related with things like neurological disorders and things like that. So yeah, I guess you don't know if the microbiome is involved at all in that crosstalk.
So I would say the microbiome may well affect, but I basically think that these effects will be indirect and therefore small in effect size and therefore difficult to capture. So as in, if you have your microbiome, the microbiome has to somehow, so I guess the metabolites of the microbiome can leak directly beyond into the lamina propria, yeah, correct? So they could be picked up directly by a sensory nerve, yeah? But then what you get from the sensory nerve is essentially like pain, I guess, right? You wouldn't get, say, anxiety, right? Whereas something like, I guess, I don't know what you're talking about in terms of psychiatric, if it's like we're talking about depression, for example, I think there would be several steps in between. Certainly from the sensory nerve angle, for you to get depression, you probably get the pain first and then you get the depression, right? Yeah. So there you might be looking more at vagal afferent information. I'm not entirely sure where the vagus where that ends up consciously, if the vagus nerve is activated, I guess, in various different sort of frameworks. And then, of course, you have the complexity that the brain sort of superimposes percepts and ideas on everything. And so that can also, then alter the circuit yet again. So it's a complex system. So I think there will be impact, but I think it will be relatively small effect sizes.
Yeah, difficult to measure.
Yeah.
Okay, we have another question. So in chronic pain sensitization, is the gut also in chronic dysbiosis? Could this be induced by treatments that perturb gut microbiome composition, e.g. antibiotics? So I guess it relates a bit with what we're talking about in terms of the potential role of the gut microbiome in this chronic pain.
Again, it certainly is true that so the sensory nerves are very, very sensitive and very good at saying something is wrong, right? So if you think about, so for example, chili, you eat a lot of chili, right? The one ion channel that we study a lot in sensory nerve biology is TRPV1. It's basically the ion channel that picks up capsaicin, which is the active ingredient of the chili pepper. So you will immediately realize, your gut will be like, yeah, you've eaten chili, and then your sensory nerves, will be uncomfortable, blah, blah, blah. That's all direct, that's very fast, that's millisecond level, right? So it's certainly true that if there are microbiota from some damaging bacterium, your sensory nerves you will know about it, right? You will experience pain, basically. Whether that, and of course, getting rid of that is a good thing. I guess it's all about sort of the subtleties. So for instance, with IBS, the complexities are that you can't really see clear all singing, all dancing inflammation. If you take a biopsy from someone with IBS, I think the reason why they diagnosed them with IBS, I may be wrong about that, but that's my impression, is because you cannot see classical inflammation in the gut, right? And so then people are like, well, these peripheral sensory nerves shouldn't be active, and therefore the problem must now be in higher spinal cord circuits, brain circuits. But even that is actually, I would say, still questionable, because actually we're now finding out a lot more about connective tissue, like fibroblasts, endothelial cells, vascular smooth muscle cells, and they actually can also talk to sensory nerves, and they can also make them very unhappy. So my feeling is that certainly from a pain perspective, if you are in pain, there probably is some kind of inflammation somewhere in your system. So either classical inflammation, where you can really see the immune system very clearly, or let's call it non-classical inflammation, where just your connective tissue is not quite returned to homeostasis, and that will send signals into your spinal cord and brain, and then your spinal cord and brain will sort of freak out and make everything worse, and like, you know, and that will add to the pain and will add to the chronicity of your symptoms. Which is, by the way, if you injure yourself and you start experiencing pain, like say you put your back out, the recommendation from a pain scientist would be to take painkillers early. Yeah, don't wait. Many of us are like, let's wait and see. No. Take it early, because you're trying to stop the central nervous system from getting sensitized. So better to take early and then stop and mobilize, right? Don't wait it out, because your central nervous system will become sort of freaked out over time.
Cool. That's great advice. We have another question.
I'm not a doctor, though, just to say.
Yeah.
You know.
But you're
from
a
science
specialist.
Perspective, you can do this experiment on yourself if you so wish. Yeah.
Also, you made a distinction between afferent and efferent pathways. Do we know whether the afferent signals from the gut are more important for conditions like chronic pain or IBS, or is it more about how the brain responds to those signals?
Really good question. So I would say the afferent signals are more important for chronic pain, or for pain in general, because it's your warning system. So when I said that the sensory nerves and the immune system together act to work for host defense, there are families, very rare families in the world, that do not feel pain because they don't have certain of these nerves. And it's a lethal condition. Most of these people die around age 20, 25, because they cannot protect their body from damage and bad things. So the afferent sensory nerves are really, really important in telling you that there is inflammation. So that's sort of the first thing. And then, yes, there can be a loop where the brain says, okay, there's inflammation, now let's activate the sympathetic nerves and shut down, or, you know, there can be sort of certain situations where it can become a loop, but the initial thing, it would be your afferent sensory nerves, yes.
Okay. Another question. So first, thanks a lot. Very mind-opening summary. One question. If one nerve, which is in connection with an inflammatory substance, what happens after that nerve is damaged? Does this lead to decrease in inflammation response, or inflammation stays because of other signalings?
Interesting. So are you talking now, when you say damage, do you mean if you take it away, and so therefore does that improve the inflammation, or are you talking about the inflammation actually damaging the nerve? I'm not 100% clear on that. I can try in a different.
I assume it's the first case, about like if you essentially take out that nerve.
Yes. Okay. Yeah. So that's interesting. So that has been done in animals, and there were different results depending on the condition and depending on the exact immune cell and the inflammation type. So sometimes taking away the sensory nerves can improve your inflammation. For example, with psoriasis, for example, there were some papers, I think, that said it improves it, and sometimes it can worsen it. So it depends, unfortunately. And I think it probably depends because of the complexities of this local inflammatory environment and all the different players and what they're saying to each other once you remove the nervous system component. But in terms of treatment, it's certainly true that very sort of traditional ways to treat pain, and they still exist, are to simply take away the nerve so that the signal can't travel up. So for example, with certain back pain conditions, they still do that. They kind of destroy the nerve that carries the information out of the bone. Like, the bone pain can be very painful, and so they just burn that nerve away, so to speak, surgically. I mean, it's very brutal treatment, if you will, to just stop the signals from coming in. So that can work, but it's complex, and of course, but it sort of depends. Unfortunately, not a very clear answer because of the complexities. Yeah.
Fine. I had some questions more from the methodological perspective. So you obviously mentioned different models and methods we can use. Can you expand a bit more on what sort of, you mentioned that using stem cell organoids does not allow you to answer certain questions, and you might prefer other types of methods. Can you, specifically in the case of organoids versus in vivo models, could you clarify a bit, like what sort of questions you could ask with one and not the other?
Yeah, absolutely. The stem cell-derived sensory neurons, which indeed you can put in an organoid, and I've done so with Joanna, what unfortunately currently you cannot answer very well, how the nerves talk back, how these afferent nerves talk back to the immune system, because they do not make CGRP. So they lack this main neurotransmitter. And the other thing that you cannot answer very well, if you're interested in chili peppers, for example, or capsaicin, they lack these receptors. They don't have much TRPV1, for example. Whereas we've used it very successfully to study cytokines in the IL-6, like JAK-STAT pathways, JAK-STAT cytokines. Most of the machinery is there, so that can be very effectively studied. So it's a bit silly. Some things are very useful, other things kind of slightly less useful. The explant experiments, so the primary afferent ones, they're great because they really contain all the different subtypes, and so you can use them for everything. It's just that there you have to be certain, well, it has to be short term, so like I said, usually over a week, and you have to just bear in mind that the communication is somewhat there's additional complexity because you have all these other cell players. And sometimes for some questions, it doesn't matter at all, right? And for other questions, it matters. But because we now know a lot, at least at mRNA level, what all these different cell types express, you can usually work out very easily mechanistically whether your particular question will be affected by that or how it will be affected. So for instance, if you're interested in ligand X or microbe metabolite X, does my microbial metabolite X cause a problem? You just need to figure out what other cell types in your dish could metabolite X affect. And then you can either say, well, I can for sure say it can only affect the nerves and therefore it has to be a direct effect, or you'll have to say, well, there is an effect, but it could be indirect via other cells in the dish, or it could be direct on my nerves.
Okay. We had a question. Do you have suggestions for possible model for in vitro vagal nerves?
Yeah, so they mentioned briefly, so the nodose ganglia from rodents, from mice and rats, if you know where to find, how to find them, that sometimes it's not so easy, but people can, there are experts who know how to take them out. You can simply take them out and put them in a dish and they will grow happily for about a week.
Excellent.
It's stem cell derived. I've had a look. I do not believe there are stem cell models for vagal afferents. There are stem cell protocols to make cranial motor nerves, which are basically vagal efferents. So if you're interested in how the vagus talks back to the gut, you can make stem cell derived vagal efferents. And we have done that before. And in fact, I think we just published on bioRxiv a protocol on that.
Out of curiosity, you mentioned chili peppers. Do we know what makes different people more sensitive or less sensitive to
yes, we do. So
food?
It's exposure. So basically what happens is really fascinating. We actually use this in the lab for experiments. What happens when you eat a lot of chili peppers is that the nerves, the sensory nerves, they know it's not good for them, so they retract from the tissue. So they reset deeper and deeper into the tissue so that you're no longer sensing it because you're no longer exposed. So the more chili you eat, the less you can sense it because your fibers are retracting to protect themselves.
Interesting.
And so we use really this both in animals. So we can actually, you can coat the animal with like a high potent, like on the skin. If you put a capsaicin cream, you denervate the tissue effectively. And you also use it for treatment. So some of you, hopefully not many, will know because it means that you're living with chronic pain. For certain chronic pain conditions, they prescribe capsaicin patches. So there is patches that you put on your skin with lots of high-dose capsaicin, and so the nerves retract, and so you no longer feel the pain.
Wow, that's super interesting.
It's a very uncomfortable treatment because actually when you first put it on, it really hurts because you haven't retracted yet. But then once they've retracted, so they're still there. They no longer go as far into the edge of the tissue.
Okay, excellent. I think we'll probably wrap it up here in the interest of time. Thanks very much, Fran, for the great presentation and answering all the questions. So we're now going to move on to the second part of the session with Joanna. So Joanna Neves is an associate professor in mucosal immunology, also at King's College London, and her research explores how the immune system interacts with barrier tissues, especially in the gut, and how these interactions shape inflammation and disease. So her work particularly focuses on mucosal immunology, and that's what she'll be giving as a bit of an overview here for this session. So over to you, Joanna.
Hey, great. Thank you so much. So yes, so Chan was saying, my name is Joanna Neves, and I'm associate professor of mucosal immunology at King's College London. So I'm going to give you a very basic overview of the immune system and mucosal immunology. And the reason I started to go with the basics is because when I attended one of the GIBA's network meeting, I realized that I was one of the few immunologists in the room. So I think it's probably good for people that don't have a background in immunology, starts from the basics, and that's what I'm going to do today. If I can move my slides. And since we are here, I'm already using my pointer, so it's ready. Okay, great. It's not moving. So I divided the lecture in three sections. The first one, we look at the basics of the immune system. Then we look at the mucosal immunology in particular, and then we look at models to study immune epithelial and other interactions, things that Fran and Alexandre were actually discussing just in the question session. So the immune system is this vast communication network of cells and chemical signals distributed in the blood and tissues throughout the human body, which regulates normal growth and development of the organism while protecting against disease. So we will have many elements from this immune system, and we will be looking at them today. So the immune system can be divided with primary lymphoid organs. These are the bone marrow and the thymus. So these are organs where immune cells develop. So for example, for B cells, they fully develop in the bone marrow, while T cells, they start, the progenitors of these cells arise from the bone marrow, then they migrate to the thymus, that is organ just above your heart, and then they fully develop there. But we also have secondary lymphoid organs and tissues, for example, the lymph nodes that you probably has heard about, the tonsils that tend to inflame when you are a kid, but also the spleen, the Pierce patches that are located in the gut, and we'll look at those today. The bronchus-associated lymphoid tissues, and also the gut-associated lymphoid tissue that we'll also look about it today. So the immune system can be divided into arms. So we have the innate immunity that is non-specific, means that any antigen that will be encountered by these cells, for example, they will respond them in the same way. And because of that, is also immediate. So, you have a response within hours. And we also have the adaptive arm of the immune system that is specific. So it means that response depends on the antigen that you get, and you have cells that can specifically recognize that any antigen that you encounter throughout your life. It's delayed, so this takes time and has memory. So what does that mean? So for example, here in this graph, you can say that innate immunity activity can strike right away after you have this challenge, and then while adaptive immunity takes a few days to start. But then when you have the second challenge, innate immunity will behave the same way. So we'll strongly respond, and then the response will decrease. While for the second challenge of the same antigen, the adaptive immunity will be able to respond much faster. It means it remembered it has recognized that antigen before and then can respond much faster. So this is actually the basis why we take vaccines. So we are training our immune system to know what are these antigens. So then when we actually encounter them, we'll be able to rapidly respond to them. So innate immunity is also not only involves cells, but also some epithelial barriers. For example, the skin that protects us from the outside is one of the skin is our major organ in the body, and it has a very important role in protecting us from the outside. But also, the epithelials in the gut, epithelial cells in the gut and in the lungs. We also have other mechanical protections like the flow of air or fluid and the movement of mucus by cilia. We also have some chemical barriers, for example, the acid in our stomach that actually kills most of the bacteria that we swallow every day with our saliva. And also the normal flora of our body has a very important role in protecting us against pathogens. So there are some key immune cells, and I will focus on very few today. So some of those are called dendritic cells, and they are very important to present antigens. So they can capture the antigen and then present it to other cells, such as T cells. We also have macrophages, and these also are able to present antigens. So dendritic cells and macrophages can be called professional antigen presenting cells because they are really good at doing this, and is how they communicate with other the adaptive cells, the T cells. The macrophages can also engulf the antigens, so they are able to do this by phagocytosis, and then they degrade it inside the macrophage itself. We also have neutrophils. They can also do this phagocytosis, and they have another mechanism that is called mitosis. So basically, when they die, they leave this jungle of DNA around them that kills everything surrounding them. We also have mast cells that release granules containing histamines, and you probably maybe heard about this in the context of allergy. Other key immune cells are B cells, so they fully develop in the bone marrow, one of the primary lymphoid organs, and they express a B cell receptor. I just realized it should be BCR and not BRC. And they are able to specifically recognize antigens, and they are known by their production of antibodies. And we also have T cells. So they're actually the progenitors develop in the bone marrow, but then they are educated in the thymus. What that means that their fully development occurs in the thymus. And in the thymus, they go through true selection process, positive and negative selection, where only the cells that are able to recognize antigens in the first positive selection survive, and in the negative selection, the cells that recognize antigens very strongly, they actually die. And the reason that happens is we don't want these cells to recognize our own antigens, so we don't want to develop autoimmune disease. So it's very important that they are educated to only recognize foreign antigens and not our own antigens. And they do that to the expression of a T cell receptor. So there are two main types of T cells. So we have the CD4, or also called T helper cells. So when they recognize antigens presented by antigens presenting cells that express this molecule called MHC class 2, and then they can recognize it. And then what they do, they produce a bunch of cytokines and chemokines that are these molecules that can gather other cells. There are many types of CD4 T cells. So some that increase the immune response, like Th2 cells and Th17 cells, but they are also, for example, regulatory T cells that are known for dampening, so reducing the inflammation. We also have CD8 T cells that are cytotoxic T cells. These recognize antigens presented by this molecule MHC class 1, and these are able to directly, for example, kill other cells, for example, by the expression of granzyme and granulus. So I'm better than explaining you this, just showing image. There is also this video that was developed by UKRI, I think around COVID, to show how the immune system develops, and I think it's quite useful. So I'm going to play it now, if I can.
Basics of the immune system. The immune system, one of the most complex parts of the human body. An intricate network of cells and molecules evolved over the course of hundreds of thousands of years to protect us from the dangers of the outside environment. So how does it work? First, detect the threat, summon help, and launch a counterattack. When a virus infiltrates a cell, for example, a cell lining our airway, the cell detects this as foreign and produces cytokines, messenger proteins, to summon help via phagocytes, a type of white blood cell, and also help to contain the local spread of the virus from cell to cell, typically the role of interferons. Specialized white blood cells called phagocytes try to destroy the intruding virus by literally eating them up. These first responders show up within minutes and make up the innate immune system. It is fast to respond, but not precise, and it does its best to shut down an infection as soon as possible. The adaptive immune system is highly specialized, but much slower. Dendritic cells collect small fragments of the virus left over from the battles of the innate immune system and ferry these to our lymph nodes, where highly specialized white blood cells called T cells are waiting. T cells are pre-programmed soldiers, each one trained to target a specific invader. When a dendritic cell carrying a specific type of virus fragment shows up, the relevant T cell will divide many times to produce an army of clones. There are two main types of T cells. Cytotoxic T cells recognize and destroy infected cells that harbor the virus, and helper T cells boost the immune response by activating B cells to produce antibodies. Antibodies are specific proteins that block the structures that viruses use to latch onto and enter our cells. Antibodies also mop up the viruses that are floating around within our body. Although T cells and B cells can take several days to move into action, they are incredibly precise, and they remember. After the virus has been neutralized and the army stands down, most specific T cells and B cells die off, but a small fraction stay alert, patrolling the bloodstream and lymph nodes for repeat attacks. If the same virus attacks our cells again, these memory cells spring into action, activating the power of the adaptive immune system without the days-long startup. This memory is the basis of how a childhood infection or a vaccination can lead to lifelong protection.
Okay, so I hope this was useful. I really like it. So now we are going to go to the mucosal side. So, and the one we'll focus today, as we are part of the GIBA, is the intestine. So the intestine has several compartments, and actually the majority of our immune cells are located in the intestine. It also has a very complex epithelial compartment that you heard if you attend the session on the first day, but I also do a brief overview today with stem cells at the bottom of the crypt, and then cells, as they differentiate, they move up the crypt and gain specialized functions. We also have mesenchymal cells or fibroblasts that have a supportive function for the stem cells, but also structural functions, and for sure very aware about the microbiota. So we have the virus, the bacteria, and the fungi that compose our microbiota, and the complex neural network that we heard from Fran just earlier on today. An interaction between these compartments is essential for the maintenance of intestinal homeostasis. And when this disruption of these interactions, you can have local disease, like inflammatory bowel disease, but you also can have systemic disease, and disease that actually may affect different parts of the body. So there are other mucosal tissues in the body, not just the intestines. So for example, we have also the stomach and the lung and the oral cavity. And the way that the immune responses occur in this are slightly different from others, because in the systemic environment, these normally contain and sterile and rarely encounters antigens, while the mucosal environment is exposed and non-sterile. So we constantly encounter antigens. So there's a co-evolution of the immune system, and these antigens that are constantly, the immune cells are constantly being exposed to. So you have an idea, this also vary quite a lot, for example, along the gastrointestinal tract. So this colonization is site-specific. So the bacteria we have in the mouth, they are different from the ones we have in the stomach, and then even in intestine, the different parts of the small intestine. So the first part is the duodenum, then you have the jejunum and the ileum. They have different bacterial composition that is also different from the colon. So all of this affects how the immune system works. So there are many different immune cells, both innate and adaptive, in the intestine, and we'll look at those in particular today. And so you have an idea, this is, the cells can be also be in different sites, and we'll look at that. So they can be in this area that is the lamina propria, but also can be inter the epithelial lymphocytes. So they call interepithelial cells. So they are called interepithelial lymphocytes. It can be also in organized lymphoid follicles in the gut. So we have a lot of the conventional cells that you can find in other parts of the body in the gut, like dendritic cells, macrophages. The reason there is +++ here is that they are the most abundant immune cell in the gut. We also have neutrophils, mast cells, B cells, and T cells. Some of these cells, subpopulations of these cells actually reside in the gut, so they are called gut resident or tissue resident cells, so tissue resident macrophages or T cells. So they mainly are there, and some are just in the lymph or in the blood, and then they are recruited to the gut in the case of infection. Then you have some specific cells from mucosal places, like the M cells that you look at in today, the intraepithelial lymphocytes, the innate lymphoid cells that are mainly mucosal tissues, can find some in the gut, some in the blood, but mainly in lymphoidal tissues like the gut and the lung. And we also have the epithelial cells. So there are several types of epithelial cells, like intestinal epithelial cells, Paneth cells, enterocytes, enteroendocrine cells, stuffed cells, and goblet cells, and the whole have different functions. So the gut has their own lymphoid structures, and the gut is the gut-associated lymphoid tissues. Those are comprise the follicle-associated epithelium that has M cells. So M cells are these specialized cells that are specialized for the uptake and transport antigens from the lumen, so from where you have your bacteria, into the underlying dome that is rich on dendritic cells. And then these cells can present these antigens to B cells and T cells, for example. We also have the Pierce patches, that is this dome over here that contains B and T cells, and is also very important for T cell priming and IgA production. So IgA is the main immunoglobulin produced by B cells and mucosal sites, especially in the gut. And finally, we have these isolated lymphoid follicles that are behind, sorry, where I can see the team's thing, so I hope I'm pointing to the right place. They mainly have B cells, and I was reading yesterday, and I didn't know this. We have around 30,000 isolated lymphoid follicles in our gut. So they are really tiny, very hard to identify, but they are main sites for B cells. While, for example, Pierce patches, at least in the mouse intestine, when you open the mouse intestine, you can actually really see these little domes across the intestine. And we also have the mesenteric lymph node. So this is the draining lymph node of the gut. In terms of where the cells are, so in the lamina propria, that is this region here that consists of loosely packed connective tissue that forms the scaffolding for the villus, as well as containing the blood supply, lymph and drainage, and nervous supply for the mucosa. Mostly we have CD4 T cells, while in the Pierce patches, most are CD4 T cells, while in the region in between the epithelial cells, so these called interepithelial lymphocytes, we have some types of cells that we normally don't find that much in other regions on the body. So these are CD8 alpha T cells and gamma delta T cells. What does that mean? So normally the T cells you hear about, the CD4 cells, Th2, Th17, regulatory T cells, they express an alpha beta T cell receptor, and we just call them T cells because that's the norm. While these T cells, the gamma delta T cells, they express a gamma and a delta chain, so a gamma delta T cell receptor. And it's still unknown what these cells recognize, and they seem to have some innate kind of phenotype in the way that they act and when they encounter antigens. And then these CD8 T cells, CD8 alpha T cells are also a special type of T cells. So the majority of our CD8 cytotoxic T cells, they express two CD8 molecules, one alpha and one beta, while in the gut, for some reason, the most of them express two CD8 alpha molecules. So that's why they are called CD8 alpha T cells. So how these cells, we have the resident T cells, but if they are not resident, how do they get to the gut? So get to the gut, they have to express this alpha 4 beta 7 that then binds to MAPCAM in the endothelial cells, so that allow us to pass from the blood vessels to the lamina propria. And then when they are in the lamina propria and they want to go into the epithelial cells, that only happens when the cells express alpha E beta 7 that binds to ecadherin expressed by epithelial cells, and that allows us to take this position between the epithelial cells. And then how do they go to the small intestine or the large intestine? Again, by expression of chemokines and their receptors. So we have cells that express CCR9, it binds to CCL25, so they will be directed to the small intestine. And then the cells that express CCR10 that binds to CCL28, they will go to the large intestine. So there's a very well-controlled gut homing of these cells. And for example, one treatment for inflammatory bowel disease, where the patients have accumulations of inflammatory cells in the gut, is using an antibody that is an anti-alpha 4 beta 7. So that blocks the migration to the gut, and so that blocks that over-accumulation of inflammatory cells in the gut. And it seems to be quite efficient at doing that. So other type of cells that we mainly find in the mucosa are called innate lymphoid cells. So as the name say, they are innate. They are mainly tissue associated, so they are antigen non-specific, and again, they are early factors with rapid and potent cytokine responses. So they can also be divided into different subsets according to their expression of transcription factors and cytokines. So we have type 1 ILCs or ILC1s, natural killer cells, type 2 ILCs or ILC2s, type 3 ILCs or ILC3s, and lymphoid tissue inducer cells. So in the gut, the majority of these cells in the human gut, the majority are type 3 or ILC3s. And these cells are known to promote epithelial regeneration, production of antimicrobial peptides and mucus. They are also known to regulate commensal microbial colonization and protect against pathogens. And they can recruit phagocytes, support regulatory T cells, and limit commensal specific immunity. I didn't go into this, but when Fran was speaking, it reminded me of something. I'm just going back. So there's a lot of known interactions between ILC2s and neurons, and there's a lot of good papers and especially also already good reviews on this interaction. I didn't went into this here, but just a caveat is that has been done in mouse models, and ILC2s are really, really rare in humans. So it's actually quite curious to know how those pathways that have been identified in a mouse setting of communication between ILCs and neurons would translate in a human setting where these cells are very, very rare. Okay, so going back, so I spoke initially how we have a very different distribution of bacteria, for example, across the gastrointestinal tract, and it's not only the strains that are different, but also the bacterial load. With the higher load that we have is in the colon, and consequently also the metabolites that are produced by this bacteria, like AHR ligands and vitamin A's and short chain fatty acids are also differently distributed along the gastrointestinal tract. And that definitely impacts the immune cells we have in the gut. So here in this very quite complex graph that because shows a lot of different immune cells, we have the intestine, so the small intestine, duodenum, jejunum, ileum, and then the large intestine. And you can see the different distribution of the commensal microorganisms with increase in the colon area, and also, for example, increase dietary antigens in the first part of the small intestine, duodenum, and then decrease over time. And this definitely influences the distribution of immune cells we have. So you can see macrophages are the most permanent immune cell and quite strong all over the place, but mast cells decrease over time when it seems that we have more regulatory T cells in the colon. So there is a very close relationship between what's happened in the part in our commensal bacteria and how our immune system is distributed throughout the gut. And also, this is also influenced in part, for example, the different presence of dietary antigens and metabolites, but also, for example, the oxygen concentrations that vary along the gastrointestinal tract. So this close relationship has been studied quite a lot, and there's several studies that point in that is something called a window of opportunity. So a critical period when early microbial exposure shapes the developing immune system. So it's not just that the distribution of immune cells varies across the gut, and that's depending on the microbiota. And we know that animals without any bacteria and virus or fungi, so germ-free mice, have issues in their immune development. So it has been also shown that this interaction between the immune and the microbial components is essential for a proper development of the immune system. And that's called a window of opportunity. So this window of opportunity is a relative short period of time that won't arise again and occurs between, just the early development and infants, so let's say up to 2 years old, and it provides a healthy benefit. So this is in the early days, the developing of the immune system is very influenced by the maternal microbes and metabolites, the maternal diet and maternal health. And then when the baby is born, the birth mode, the nutrition, the genetics, and the infection that might have really shapes the microbiota and shapes their immune system. And that will influence the adult. So when these infants have, good nutrition, they don't have a lot of infections, they don't need to be treated with antibiotics early on. They can develop this eosbiosis or diverse microbes, and that drives a healthy immune imprinting, and they can develop healthy immune system that they'll impact their health at adult stage. But if opportunities are missing and there is this, they have infections, they have to be treated in the microbiomes. If it's known that, for example, breastfeeding and vaginal birth is very beneficial for this health imprinting, and if that doesn't happen, then it could have a pathologic immune imprinting, so that the changes and the dysbiosis that occur at this early stage can really shape how the immune system develop, and that could influence how then in the adult stage people will have been more susceptible to certain infection and inflammation. So moving from that on, the epithelial compartment, I said I will revisit it early on. So we have a lot of different types of epithelial cells. So at the bottom of the crypt, we have these stem cells or LGR5 stem cells, and these cells have the capacity to become any type of other cell. And as they develop, they move up the crypt. And they can also become Paneth cells, and these cells are very important because they secrete antimicrobial peptides, so keep our bacteria at bay, but they also have important supportive roles of the stem cells. Then we have enterocytes that have absorptive function, enteroendocrine cells are quite rare, very important in hormone secretion and very important in interaction with the neurons. We have Tuft cells that produce, these are called the sensory cells that produce IL-25, and we have goblet cells that produce mucus. So there is a mucus layer between our own cells and the microbiota. So in the intestine, we have this, the the small intestine, we have the Paneth cells here at the bottom of the crypt, while in the colon there are no Paneth cells. So the support of that is given to the stem cells by the Paneth cells in the small intestine is given by fibroblasts that's surrounding here the crypt. The other difference between the small intestine and the colon is the colon has way more goblet cells than the small intestine and has two mucous layers, so inner mucous layer and outer mucous layer, while in the small intestine we only have one mucous layer. So there's some difference. So we call it the intestine, but they are actually two organs with different characteristics, so the small intestine and the colon. So when we want to study what happens in interactions between the epithelial cells and the immune cells and the neurons, there's always a trade-off between the experimental control and the physiological structure. So we can use, for example, cell lines that give a lot of experimental control. Our ideal is we do these studies in humans, and nowadays using, for example, single-cell transcriptomics and spatial biology, you can get a lot of information, but it's still hard to do mechanistic studies. So one of the ways of going is, for example, using intestinal organoids that are the sweet spot in the middle that give you some experimental control and some of the physiological structure. And if you think you want to understand human biology, maybe intestinal organoids could be human intestinal organoids could be more useful for that than other approaches that have been traditionally used. So what are organoids? So they are defined as these three-dimensional clusters that are grown for pluripotent or adult stem cells that spontaneously organize into organ-like or tissue-like structures that are enriched for the cell types typical present in the tissue of origin. So organoids were considered the method of the year in 2017, so that's almost now 10 years ago. But the use of non-animal methods, and a lot of those are organoids, has been exponentially increased over the years, and it's something that has been quite, there's quite a push from the government and other associations to drive this development. So there's a multitude of organoid models from human tumor-derived organoids or human tissue-derived organoids. And nowadays, so the first ones were the intestine, and nowadays you can make organoids for anything you can think about. But today we'll just look at the intestine. So to make organoids, you need a cell source. Those can be induced pluripotent stem cells or tissue stem cells that you derive from tissues, for example, from patients' biopsies or resections, or from animal models. You need some soluble factors that are essential for the growth of these organoids, and you need a matrix where these organoids grow that is like a gelatin that allows them to grow in 3 dimensions. So for intestinal organoids, we can make them by isolating crypts for patients' resections or biopsies, or from mouse tissue. We could isolate just the LGR5 stem cells. This is a bit hard to do. You need a special mouse model that label these cells, and the efficiency when you start with a single cell is quite low, is 1 in 10, while when you start with a crypt is much higher. And it's because the crypt, in addition to the stem cells, also has the supported cells like the Paneth cells that allow their development. And you can also use induced pluripotent stem cells. Then you culture them in precise conditions in this matrix. The matrix mostly uses called matrigel. And then they develop into this 3-dimensional structure that has all the type of intestinal epithelial cells present in the intestine. And the cells maintain the apical basal polarity. That means that on the outside is where you have your immune cells, and inside is where you have your bacteria. So these some pictures taken many years ago in my lab, where we have the crypt at day 0, where it's like open structure, round but open structure that closes by day 1, and then you start to get these invaginations by day 5. So I have this video that you can see that the organoid developed over 7 days, started this round structure, and then I say develops this beautiful flower-like structure over time. So we can make these tissue-derived or adult cell-derived organoids or induced pluripotent stem cell-derived organoids, and the organoids you choose depends on the question you have, because they have different characteristics. So the tissue-derived mimics the cell stage of the age of the tissue you got it. So if you got it from a child or from adult, you'll have organoids that mimic the children's intestine or the adult intestine, while induced pluripotent stem cell organoids always mimic the fetal stage. Nowadays, we can add some molecules to these organoid cultures, and that allow us to further maturate them so they look like adult-like, but not still adult yet. Also, the cell composition is different. So the tissue-derived organoids, you only have a layer of epithelial cells, you can see here, and also this picture from my lab, while in these pluripotent stem cell-derived organoids, you have the epithelial cells, and then you have a layer of fibroblasts surrounding it that co-develop with these epithelial cells. So if you want to do some studies that you want to look extracellular matrix production, for example, on the role of fibroblasts interacting with epithelial cells, then maybe using induced pluripotent stem cell derived would be a better option. Other things to have in consideration is the variability is quite high when you use tissue-derived organoids. The patients, you get the samples from the patients that have, different age, exposed to different drugs, different diet, different microbiome. But, if you want to test a new drug or a new therapy, you do want that variety because it's very good to test in a diverse population. While when you induce pluripotent stem cell derived organoids, you can always use the same cell line, so the variability, and then they have been extensively characterized, most of the time sequenced, so you reduce the variability. If you want to do genetic manipulations and, for example, knock out a gene of interest, it's easier to do it in the setting of induced pluripotent stem cell derived organoids because you can do CRISPR at the stem cell stage and then make the organoids, while you can do it in adult tissue-derived organoids, but you'll be CRISPRing adult cells, and so it's a bit difficult, but still possible. So it really depends on your question and on the tools that you have available in the lab, what type of organoids you'd use. So we know from what we heard in this lecture that the intestine is very complex and has way more than epithelial cells, and also have the immune cells that we spend so much time speaking about, but also the neurons and the microbiota. So organoids are a great model, but they don't have this. So that's what my lab and others have been doing for a long time, is to add complexity to these systems. And for example, we have a lot of studies where we add immune cells to study immune epithelial interactions, but we can also add bacteria, metabolites of food allergens. We can play with extracellular matrix where the organoids grow, and we can add neurons. And that's what we'll be looking today, is just a little bit of detail. So to add immune cells, you can, or immune components to the organoid culture, you can just add the cytokines that are the molecules produced by immune cells to the media and see which effects they have on organoids. You can also just add the immune cells to the media, and some would penetrate the matrigel, some don't. So this is, I don't think, the best. Most of the time, this is not the best approach. Our favorite approach is put everything together. So we embed the immune cells and the organoids in the matrigel. This allows direct cell-cell contact between immune cells and epithelial cells in case the mechanism you are studying requires that contact. And then you want to see if that stops, and this is just a picture of one of the organoid in the lab surrounded by green immune cells. So if you want to see if your mechanism was contact dependent, then you can separate again. The organoids can stay at the bottom of the transwell embedded in matrigel, and you can have your immune cells at the top. You can also, if you want to add bacteria, you can microinject them into the lumen of the organoid. It's be like an IVF approach. It's feasible, it's quite technical, complex. You can also invert the polarity of the organoid, so then you have the apical out. So then if you add bacteria, they will be directly interacting with the apical side of the epithelial cells. This is done, when you do this, you have to treat an organoids and they are in suspension, so the viability is quite low, and not all organoids invert. So you have some that are inverted, some not. So it's still, I would say it's a protocol that still needs to be optimized by the field to be able to use it quite confidently. I think the best way, if you want to study both interactions with immune cells and bacteria, to use this transwell approach. So basically, you still grow the organoids in 3D. They still have a different type of epithelial cells, and then you break them and you plate in the transwell. So you have this 2D organoid, and then you have easy access to both the apical and the basal side, so you can have the bacteria to one side and immune cells to the other. So this allows us for this interaction. So in terms of adding neurons, I'm going to mention two approaches where they use human iPSC-derived intestinal organoids for neuron co-cultures. One has been published many years ago from the James Wells lab, and after they develop them so these are human iPSC-derived organoids co-culture with neurons, and after they do this co-culture, they need to transplant under the kidney capsule of the mice for fully development. And with Francisca, then we have this brilliant PhD student, Noel Chiong, that we developed this similar similar protocol, but adding other molecules to the protocol, we don't need this transplantation into mice for fully developed. And more recently, there's a new protocol where you use iPSC-derived Tesla organoids that by adding some molecules to the media, these organoids already develop with neurons, and this for fully development also requires transplantation into mice. So just an overview of the most recent protocol. So basically, this is something we do when we grow iPSC organoids. We start with iPSC cells. We treat them with molecules that drive the differentiation into endoderm, and then further drive the differentiation in hindgut, so the primitive gut, and then we embed them into these spheroids and they make organoids. So this lab has, by Jensen Spence, has seen that by adding epireglin to this stage, they could have a fully developed organoid that also contains neurons. For that, after they have the organoid developed, they transplant under the keep-like capsule of the mouse, and they let it grow for 10 to 12 weeks before they analyze. And when they analyze these organoids, they could see, so for example, this image here in yellow, you have epithelium marked by icadiran, and then other images you can also see develop of neurons, looking at tubulin, endothelial cells, looking at PCAM and smooth muscle cells. So it's an organoid that has many different components. So while the conventional organoids only have the epithelium and the fibroblasts, in this enhanced epireglin organoids, you have intestinal epithelial cells, epithelium, mesenchymal, smooth muscle, endothelium, and neurons, just by adding this molecule and by transplanting into the kidney capsule of the mice and waiting 10 to 12 weeks. So the initial protocol, that is the one we modify, is a co-culture, means you start by growing your organoids and growing your neurons separately, and then you join them when the organoids are day 7 and neurons are day 14, and then allow it to develop together for the rest of the time. And what we've seen, and as we can see, develop of, again, glia cells and neurons in these organoids, and you can see here how they are actually directly interacting with the epithelium. So the initial protocol requires these organoids to still be transplanted into a mouse for fully development, while the one we are working on at the moment does not require transplantation. And we want to further develop this to actually other elements. So, for example, we can challenge these organoids with cytokines or add immune cells to see how they interact with the fibroblasts, the epithelium, and the neurons. We can also, for example, add microbial metabolites or just supernatants from some microbial culture to see their impact. We can add other type of neurons. So, for example, as Fran was speaking, vagus efferent neurons could also be added to this organoid. So the enteric neurons, vagus efferents, and organoids could all be differentiated from the same line and then joined for this co-culture. So the key take-home message from this session today is that the immune system has innate, so rapid and non-specific and adaptive arms. So they are delayed, specific, and with memory that can interact with each other and very important for these immune responses. The intestine is very densely populated by immune cells that can be in the lamina propria, interepithelial cells, in the gut-associated lymphoid tissues. The immune apparatus varies along the intestine and it co-develops with the microbiota. And this interaction between immune cells and the microbiota is required for proper immune development. And this is called, especially in the special time between early age and 2 years old, is called a window of opportunity. And intestinal organoid co-cultures with neurons and immune and microbial compartments can be used to study these interactions in the gut context. Hope you found this helpful. And I know this was a lecture, but I actually use data from quite a lot of people. So I just I'd like to acknowledge my team, our collaborators and funders. Thank you.
Excellent. Thanks so much, Ivana. That was really a nice presentation. I'm sure people will find it super useful, especially me. I don't know much about immunology and learned quite a lot. So great job.
I also learned quite a lot preparing it. So.
No, but it's great. It's great because the lectures are recorded as well, so you can kind of look back, and even though it's a lot of information, you can then go back and actually look at it and digest a bit more. So this is great. We have a few questions already. There's one with already 4 thumbs up, so I'll start with that one. Do you have any recommendations on the types of immune cells or cytokines to investigate when assessing the general effect of a diet in mucosal immunity?
The true answer is no. I don't know. So what we normally do in the lab, for example, if you were saying you want to study an inflammatory environment or non-inflammatory environment, I could advise you some cytokines to add. But to be honest, in terms of diet, it's something I'm not familiar with. So I don't know what would be the context that would be done, to be honest.
I guess it would be probably an indirect effect where if the diet affects the microbiome, you could use specific models to test those specific bacteria affected by the diet or something like that.
Yeah.
Yeah, maybe it would be more, yeah.
So it's an honest answer. I don't know. I don't think I know enough about diet and to be able to assess that.
That's fair. That's fair. Okay, another one. Thank you for your talk. Very interesting. There has been a long-standing problem with co-culture of in vitro intestinal models with anaerobic gut bacteria, for instance, Clostridium, as the environment is too aerobic. Does the organoid model overcome this problem?
So it can overcome in some ways, and some ways it just hasn't been a problem. So there are some approaches that you can use, like you can have organoids in fluidics devices or in chips, like this organon chip, and in those contexts you can actually control the oxygen concentration in the different chambers. So the chamber would have your bacteria, and the chamber you have the epithelial cells, so they can have different oxygen concentrations. So that allows you to then use anaerobic bacteria. I have to say, for example, in the lab, we had a project many years ago that didn't follow through with Fusobacteria nucleatus, that also anaerobic, and we thought was going to be a big issue. So what we did, we culture foods with different oxygen concentrations and the organoids at different oxygen concentrations, and then found one that both were happy with, and then went forward with that. So that's another approach. But using some special devices where have different chambers where you can use control oxygen concentration in different chambers, differently approaches that people have using quite a lot now.
Okay. I have another question from Martha. Thank you for such a clear, interesting talk. I'm not familiar with gut organoids at all, so I'm curious, do the more complex intestinal organoids naturally contract, even without muscle tissue? I think that ECs can also show contractibility. So does it affect the experimental setup? Do you inhibit contractibility, or is it so minor that it's negligible? Or perhaps stimulating contractibility would enhance physiological validity? Thank you.
So the tissue-derived organoids don't have any smooth muscle cells, so they do not contract. Again, if you put them in these organon chips, you can induce contraction on the chip, so then they would force contraction on the cells. If you use iPSC-derived organoids, they do have these smooth muscle cells, the normal iPSC-derived intestinal organoids, nothing fancy. And I didn't know they contracted, and then until we did these co-cultures with neurons and then challenged these co-cultures. I think I don't remember, but we did some calcium imaging on this. And while we were doing, we look at the screen and the organoids were contracting. So it does happen. So the neurons were being activated and inducing the smooth muscle cells to contract. So this contraction can happen. It's something that hasn't been study a lot except in the organon chip models, but it's something that you can induce if you think it's important.
Okay. Thank you. I have a question about whether you see your organoid models being used to study how the gut epithelium and immune cells might influence gut nerves or even vagal signaling.
I hope so, and I hope the reviewers will agree with me at some point. I think so. I think this model still has a lot of room for improvement, but I do think it is a model that will allow us, like Fran was giving the talk, and she gave really great examples of this interaction. But what we know is mainly from mouse models, so we really lack human models to understand what's happening. And obviously, and especially, like it's very hard, as Fran was saying, to even isolate these neurons from human tissues. So I think using iPSC organoids and iPSC-derived neurons is a way to overcome that. And as Fran said, there are limitations, and you need to be aware. We spent more than a year optimizing our enteric iPSC-derived enteric neural protocol because we didn't get, the markers that we expected for internal neurons, and that took years to, have a protocol we were happy with. So still, getting there, but I think it will be a way forward and allow us to study this interaction, especially in the human context, that we cannot do another way.
Great. Given the early life window of opportunity, what degree of immune plasticity has been demonstrated in adults at the cellular and epigenetic levels?
So there's a lot of plasticity in immune cells. They for example, CD4 T helper cells, they can become each other quite easy. So you have these naive cells, and depending on the stimuli that they receive, they can become different type of T helper cells that produce different cytokines and can be pro-inflammatory or can be regulatory. There's a lot of plasticity, but it has been shown that if you miss this window of opportunity in early development, and again, there's some studies in humans, mostly in mice still, that then you are really more susceptible to further effects, for example, to IBD, to diabetes. So there's a training that seems to need to occur early on and seem to have some level of epigenetic regulation that you cannot, if you miss that training, you cannot do it afterwards. So it seems that this early age it's very important for the proper development of the immune system.
Mm-hmm. So building upon that, a question from me. So could you expand a bit more on during this window of opportunity, how our immune system is able to distinguish and learn which bacteria are commensal and which one are harmful? What actually happens to build that sort of immune recognition?
So it's early on we have it's more kind of our adaptive immune system is still kind of developing. It's more an innate response, and even the cytokines that infants, not neonates express is very different than the infant cytokines that even as children 5 years old will express.
Mm-hmm.
So there's a really training of the immune system it's somehow not over responsive that early on, so it allows for this colonization to occur without attacking this it's a naive system that has this huge amount of colonization happening really fast, and the immune system allows for that to not be overreacted. So it's still able to produce some pro-inflammatory cytokines like IL-8 very early on, but for example, interferon gamma production is quite low to start with, and even the cells that you have are different. And if I'm not wrong, I think even the expression of the TLR, so these molecules that can recognize bacterial signals, is quite different in this early stages than it will be later on. So there's all training that is happening in this co-evolution that is quite interesting.
Yeah, that's very cool. You talked about the distribution of immune cells and microbiome differences along the intestinal tract. I was wondering whether we know the cause and effect in this situation. So is the immune cell distribution different because we have different bacteria, or do we have different bacteria because the immune cells are different?
I don't know. So I know there are some studies. It's funny, I don't know about immune cells, but I do know about neurons. So there's this, no, because there's this very interesting study that shows that the neural distribution across the gut is also different. And it's very clear, and I didn't know this, but it seems to be very clear, different regions also. And then they did this in germ-free mice, and there was absolutely no difference. They were the same throughout the whole intestine. So that seems to be driven by and I also know the same is true for these TLRs, for these receptors expressed by immune cells and also by epithelial cells, that there is a different distribution of TLR expression throughout the gut, and that's completely abolished this difference in germ-free animals. So it seems that the microbiota is driven of the difference over there.
Interesting.
So on that sense, I would say is microbial driven, but I don't know. I would assume there's some studies doing the same in germ-free mice, but I just don't remember what they found.
Yeah, but I guess in humans it would be very difficult to really pinpoint the cause and effect.
And sampling in different places would be very hard.
Yeah, exactly. Have another question. Thank you for the amazing talk. Given the enteric nervous system and the gut-associated lymphoid tissue are in constant crosstalk, how much of the brain signal is actually just a downstream effect of macrophage polarization in the lamina propria?
I don't know.
Some difficult questions.
Yeah, very hard questions.
We have all experts
I
in the
definitely
audience.
think there is an impact of what happens in the gut from what we have reactions in the brain. So Fran didn't mention, and I also didn't mention, but one of the reasons we did this co-culture of neurons with organoids is we want to study Parkinson's disease initiation in the gut. So there's this idea that there is actually early events that occur in the gut of Parkinson's patients that might be driven of the symptoms they find later on. So Parkinson's disease patients have gut issues way before they have any motor symptoms, and there's also a lot of some genetic overlap, for example, in Parkinson's disease and inflammatory bowel disease. And there's some mouse studies that actually shown that alpha-synuclein is the protein that forms aggregates in the brain of Parkinson's disease patients, that if they inject misfolded alpha-synuclein labeled in the gut of the mice, then they could find it in the brain. So it can migrate to the brain. So our hypothesis is maybe some events in the gut, perhaps inflammation, perhaps changes in the microbial composition, because again, Parkinson's disease patients have microbial dysbiosis, drives this misfolding of alpha-synuclein in the gut, and then that is an early event that could migrate to the brain and initiate the process in the brain. So I think there's a lot of this crosstalk happening that we are not aware. And again, that's all. The mouse study seems to be very clear, but the human ones is still hard to mimic this and see what's happening.
Have another question. Will macrophage polarization in the lamina propria be the early stage to triggering the mucosal immunological response?
Could be or not, could be other early stage. So for example, even in just disruption of the barrier, it could be one of the early stages that disrupts, that leads to immunological response. So if you have this, normally you have this tight layer of epithelial cells, and bacterial metabolites would not go through. And if you have disruption of this epithelial layer, then they could much easily go through and activate the immune cells. They could definitely activate macrophages and polarize them to be more pro-inflammatory, but they also can, for example, activate innate lymphoid cells that are there and ready to act and express a lot of cytokines. So it's really hard to know what's the early stage of any of these processes. They are very complicated. I do, for example, teach about inflammatory bowel disease, and I always say, we try to understand this is the order, because in our head makes sense that you disrupt the barrier and then you activate the immune system, and then but we have no idea what happens first. So you could have a pro-inflammatory immune system that, because of that, produce so much cytokines that disrupt the barrier, for example. So then that would be the first event, and barrier would be the second.
Excellent. Could you suggest approaches to study direct interactions of microbial proteins with immune cells or nerve cells?
I think you could just incubate them together. You can, so you can just incubate metabolites with immune cells or nerve cells and then look at what happens to each one of them. So you can look at the cytokines these cells produce, or the mediators produce them. You can use ELISAs or flow cytometry or Western blot if you have enough cells. So I think you can just incubate them directly and then see what happens.
Excellent. I guess I can ask one last question out of curiosity. So there's an increasing interest in the gut microbiome field of going beyond just bacteria, right? Doing multi-kingdom analysis and things like that. Do we see general trends in immune recognition responses between, let's say, bacteria, viruses, fungi? Do we see differences between these broad groups, or is it always very specific or hard to really?
So, for example, viral response is mainly CD8 T cell mediated. Mm-hmm. So it's very, so there's a big difference there. While bacterial response just does not tend to be mediated by CD8 T cells. So there's a big difference. I have to be honest, I don't know about fungi, and I don't know if it has, so at least the basics of the response we don't know. I think the response is very driven by Th17 cells, for example, against fungi. For example, there are things that are very clear. For example, helminth infections, they drive Th2 responses. And it's like, if anyone's studying Th2 response, they do helminth infections because it's a clear cut of what's happening. So it's very nice and specific. Other is, I forgot the name, SFB. SFB, yeah, it's a type of bacteria that and is known that it drives Th17 responses. So there are some classic things that immunologists use them actually to study specific responses, and then virals is CD8 T cells. But I'm not sure I could say it's for all. So I think it's really can be very specific, and some are very so clear cut that we use them as models, and some I think probably involve many different types. So it's more complicated to study.
Yeah, excellent. Well, thanks so much, Joanna. Thanks, Fran and Joanna, for these great talks. I hope everyone found them super useful. So this really wraps up all our lecture series on the gut, immune, and brain axis. Thank you, everyone, for joining us. So as I mentioned in the beginning, the recordings will be made available on your YouTube channel. We'll try to do that as soon as possible. They'll need a bit of editing just to make the videos a bit easier for upload. So yeah, thanks so much for everyone, and do let me know. I assume Joanna and Fran will be open to getting any other questions or answering anything else
happy to
by email. Yeah, so if you have any further questions, feel free to reach out. So yeah, thanks everyone, and I guess see you all soon. Okay.